This is really a advertisement for collaboration in Australia for Australian people who are doing research. Now, Yolita's story is very similar to what's happening in Australia. Never had a never had a research scientist based in Australia that studied myxos ever. There have only been curiosity-driven souls like myself who have added to the knowledge of Australian myxos. Some of you would have been at the last ICSM and you would have heard from Sarah Lloyd. And then there are a few others on the East Coast as well. And so there's only very few of us studying slime molds, and we are trying to lodge our specimens at Herbarius, get the information onto GBIF, etc., so that people understand what Australians have got. The problem in Australia has been the fact that it's taken me a long time to realise that I'm not the stupid person that I was, not being able to identify slime molds. I now realise that slime molds do not fit the books that are produced in the Northern Hemisphere. And I have spent many years, well, not many, I've only really been studying these for 4 or 5 years, banging my head against a brick wall, trying to figure out why could I not identify material in Australia? And I now realise it's because it's not what you all see in the Northern Hemisphere. So this is really my story, but it's also a story of other Australians studying slime molds. And that is that when my specimens are lodged at the West Australian Herbarium, of course, the information eventually gets to GBIF, and you end up with a distorted distribution like this one up here, where Licea grows in a straight line, okay? Licea pygmaea. Also, excuse my pronunciations. Some of these genera I've never heard. Wait till you hear about the Arceria. I've never heard, so I have no idea if I'm pronouncing them correctly. Australia, I'm here to sell Australia because I want collaborators. Huge continent, long separated from the rest of the world by an ocean. So for about 50 million years, our own unique vegetation has grown. So about 70% of our species are endemic. This means that we must have a very endemic myxoflora, surely, and our own assemblages. Not to mention it's large, it's an ancient land, very poor soils. And also we have the climate regions go from tropical in the very north down to the cold forest areas in the south. So we have everything. And on the, get the little pointer out, on the southwest here, we have a biodiversity hotspot. We also have a diverse biodiversity hotspots up the east coast here as well. So we have a lot, I feel we have a lot to discover, and we need your help. As you can see, no specialist myxo researchers. Now we have had 330 approximately species recorded from Australia, but many of them are only 1 or 2 specimens, so they're possibly incorrectly identified. And we only have 10 approximately new species, and I think that's actually a very low number compared to what I think is here. So why does Licea pygmaea grow in a straight line? Well, it's because it's due to a camping trip in Australia where we left home here on the west coast of Western Australia. And we took a camping trip where I collected substrate because I love doing moist chamber from arid areas. And this is my hometown here. And we took a trip to Uluru, and that's the beating heart of Australia. I'm sure you've all seen this beautiful image of Uluru. This is the Great Central Road. This is the road we took, dead straight, gravel. Here, this is all we take with us is one vehicle, and all our tent and camping gear, etc., is in there, including, much to my husband's frustration at time, a lot of space to collect substrate. That often goes on the top of the vehicle. But here we are helping a couple of locals who had a flat tire. We came across them one morning, they'd been there all night. Just notice the vegetation. Now, this is a very arid region, arid area of Australia. In fact, 70% of Australia is very arid. But when we talk about arid, we're not talking about unvegetated. There's stacks of vegetation in Australia. There is no sand dunes and that sort of thing. All this vegetation is and I'm just going to have a mental blank now, but it's, what's the word? Anyway, it's It's Australian, so it's used to the conditions. Now, just there, you can see some spiky grass, which we call spinifex, and then just some low shrubs, quite sparse vegetation. The shrubs are proteaceous or acacia. But have I correctly identified Licea pygmaea? Now, who knows, because I have never seen any real material. I don't understand really, when I was first starting to study slime molds, exactly what was Licea pygmaea. It was supposed to be not that common, but actually when I look on GBIF, there's lots of Licea pygmaea. Okay, so these are my images. I don't do photo stacking, and I hope that you can see what I can see. But just they're immature here. The one thing I want you to notice there is the interrupted platelets. So this is a Licea that's supposed to have platelets, but in Australia, and maybe Licea in the Northern Hemisphere as well, the platelets are somewhat interrupted. The spores are usually quite dark. The picture there hasn't come out so well. So that's what they look like just before harvest. I've had some SEM done. And I guess, again, you can see that the plates are interrupted. They don't often go all the way across. So in fact, how they normally dehisce is via a circumscissile dehiscence. They tend to the top just tends to fall off, okay? It's got the minutely watered inner peridium, and then the spores are very watered. And also they have the thicker spore wall on one side, okay? Now, all correspondents will have a chat afterwards, and you can come and tell me whether I've got the ID correct or not, okay? That Licea pygmaea grew on substrate that was collected at Warrakurna. Here, there's Boorloo, Perth on the left, and there's Uluru, and it was collected on substrate in that area on what's called desert she oak. It's Allocagerina decaisniana. Beautiful tree, woodland tree. But in fact, the immature trees look more like a feather duster sort of growth habit. It's not until they're adults that you get the tree. They have a very thick bark, and that's to protect them from the fires, the normal fires, not these terrible climate change fires, but Australia is a country of fire. Fire goes through quite regularly, and it's to help protect the bark of the tree, or the bark help protect the tree. Also, as harvested from the same substrate was kind of Stelium apitectum, Comatrica ellie, Stemonotopsis, and two more species. Now, one of them is this one, which is Echinostelium porciphyllum. This is apparently not that common, but I thought this was really cute, and it was very obvious that this is what it was to me. The only thing is it's a little variable from what I normally read in the textbooks. The spores are a little smaller than typical, and they were quite a lot taller. This was the first record for Australia, and that was on that same substrate. Also on the same substrate was this little guy, Arceria porziaca. How do you pronounce it, folks? Very rare, apparently. Well, when you look on GBIF, there are no records at all, but of course there are records around. So this is proving extremely common in Western Australia. I've collected it up in the Kimberley, in the middle of the desert, and certainly around the Perth area, you'll find it quite frequently. Always grown on moist chamber, but it's very, very common. So that's just showing you the sorts of things that we're finding in Australia that are different to the Northern Hemisphere, but also variation as well. So I do a lot of moist chamber because that's my favourite thing to do. And the other thing is that collecting from the arid regions means that you get some really good fruitings in moist chamber. Usually you can get 6, 7, 8 species just growing in the one moist chamber, and that's great. You can get up in the morning, go and have a look in your moist chambers. Haha, look what I found. I find field collecting a pain in the neck because you have to get down on the ground and you have to look for things, and it can take a long time. So, yeah. That's not my favourite thing to do. So I'm now going to talk just a little bit about some field collections I've been doing and then what I've been finding. So, that's the capillicium of the Arceria is so gorgeous. I call it the chubby capellicium because it's quite chubby compared to most of them, but that's what it looked like. So just near where I live, and I live right up there, there's my home up there, and just 200 metres down the road is this tiny little 10-hectare reserve. It's the Swan River Plain has a lot of freshwater wetland areas, and they're just completely surrounded by suburbia now. So my little local wetland is Blue Gum Lake, and then nearby is one called Burragoon Lake. So I started looking in Blue Gum Lake because I don't know, some of you obviously on the Slime Mold Appreciation page. Peter McDonald is on there quite a bit too. And we were having a chat about Trichia decipiens, which is now Hemitrichia decipiens. So I went down to the Blue Gum Lake, and this is what it looks like. It's very cute. It has fringing melaleuca, which is then followed by Eucalyptus rudus in a transitional zone, and then after that is Banksia woodland. So we were looking at Hemitrichia or Trichia decipiens, and the Trichia decipiens is well recorded as occurring in Australia, but Peter and I were very confused because it was not matching the texts at all. So here we are, we have the usual colour for Trichia decipiens, what I call a bleb here. And I know they can be white, and they turn a brownish colour. But the thing you need to notice about this one that's just before drying out is it's got this clear circumscissile dehiscence. Not that it dehiscence at that point, it actually explodes out the top, and then you end up with an edge, entire edge. And this is what they end up looking like. And that's fine, except that's not really how it's well described in the texts. It's the spores that really killed us. We cannot figure out, is this really Trichia decipiens? Okay, so every single text in the last 150 years has said this is Trichia decipiens. So when we look at this and compare notes and we go, there's something we're missing, we must be a bit dense or whatever, we can't figure out what this is, okay? The spores are gorgeous, absolutely gorgeous. So here we go. So here already got people saying, no, this is not Trichia decipiens or Hemitrichia decipiens. Okay. Is it Trichia crateriformis? No, it's got some features of it, but the spores are completely wrong. Okay, so I did quite a lot of research in this area. So this is the Banksia woodland. That's the sort of substrate it grows on, just dying Banksia. And while studying this Trichia decipiens and collecting lots of specimens from around Perth, and they all look the same, on this sort of substrate, then I found this. Now, this little guy starts off with white blebs. I thought, oh, well, Trichia decipiens can start with white blebs too. But they turn an olive colour, and this is just prior to harvest, a really lovely deep green. But you can already tell it's subsessile. That said, that stalk has the spore-like cysts, as does Trichia decipiens. So it does have some cysts, and it has a bit of debris in there as well. I found this at a number of sites. Now, the funny thing was that I couldn't get it to dehisce. So there's no circumscissile line of dehiscence at all. And it's quite tough. And I actually had to pull the damn thing apart with the forceps. And I thought, what's going on here? And then, okay, so let's just look at the capellicium. The spores are just watered. But what it's got is it definitely has a double peridium. You can just see it splitting there. And I've seen it under transmitted light as well. Cannot key this out anywhere. This has got to be a new species. But also, whilst looking in the same bushland, I came across this little guy. And it looks a bit like Trichia botrytis, but it's not. And I have been talking with Carlos Lardo about this, but of course he can't really help much at the moment. I sent him some material and he did some SEM, but the problem was that this species is so little that a lot of the capellicium is immature or rudimentary. And we really couldn't get a grip on what was going on, but he did eventually get a bit of SEM and said it's got spiraled ornamentation, because I thought it was a Perikina. This is just some more. This is from Blue Gum Lake, from the Burragoon Lake. So it's just shows the different variation in patination. So it only grows in this Eucalyptus rudus bushland. Eucalyptus rudus is a flooded gum, so it occurs around lakes and waterways. This is actually, they're usually large trees, but there'd been a bushfire down there about 12, 14 years ago, consequently a lot of regrowth. So I was collecting other slime molds in the leaf litter and brought them home and found this little guy sitting on a leaf and thought, oh, that's cute, Trichiobotrytis. That was a year of Trichiobotrytis for me, because it seemed that everything we found had that sort of giraffe patination on it. But this little species only grows in the leaf litter. And so that's why I've got this little picture here of a couple of logs and deep leaf litter. So the leaf litter has pushed itself up against the edge of the logs. It's got to be quite deep. It's got to be sort of almost on the ground, the leaf. Otherwise you won't find it. And it will only grow on the flooded gum. It can have the leaf, the flooded gum leaf. You can have a Banksia leaf in there. You can have sticks, you can have twigs and bark. Doesn't grow on any of that. So it's only on the leaf. So I have a feeling that this is probably very host specific, and obviously will only occur in Western Australia, where flooded gum occurs in the southwest area. So this is a bit of SEM for the little guy. So it's got this, you can see the the yellow bands. I don't know if it's got a double peridium or not. I haven't quite figured that out for myself. Always ends up with a bit of a base left on it here, always stalked. This is the capillicium, so you can see the spiraled ornamentation here, but they're a bit broken up, they're a bit interrupted, a bit loose, not always there, but also Carlos and I trying to decide whether it's a Perikina or a Trichia. So the minute he saw this and it's got the spiraled ornamentation, he said Trichia, but he said the problem is it looks like Perikina. So this is something that needs further investigation molecularly. It's got cute capitate ornamentation on the spores. Here we go. So that's that little guy. So that's the end of my talk. So really, I just want to say that those of us who are all self-taught researchers are finding all these really interesting material in Australia. The one thing I would probably say is the red tape to get specimens in and out of Australia is tricky, but it can be done. And when you start research, when your students are starting some research, and we would really like to see Australian material included in all the work that occurs in the Northern Hemisphere now, and that we are down here, we're willing and ready to help, but we also, to reciprocate that, is to be able to describe some of these species. I'm going to need a bit of help because I don't have access to, we don't have access to things like critical point drying. We can do, some of us can get some SEM done, but also we don't have access to molecular work, and we wouldn't understand the results anyway. So that's the end. Thank you very much for listening. Runner. Thank you. This one up there. Oh, Arena.
Thank you, Karina. Wonderful talk.
Thank you.
And I'd like to comment a little bit about Arterias. It happens that I received some material from you and from other Australian citizen science and researchers. And it's indeed like every time I get the Arteria from Australia, there is, when I build a phylogenetic tree, there is clade for, let's say, temperate Arteria ferruginea and Australian Arteria ferruginea.
Right.
So it's almost never the same species as the European, or even with even if like the other tropical clades that appear on the phylogenetic tree. So it's really a Klondike for undiscovered species diversity. But it's not the case with the Arteria pausiaca. It happened to be the exclusion, and it happened to be not on the Australian phenomenon. So it's a nice species in Arteria pomiformis group. And yeah, it occurs not only in Australia, but in tropics as well.
So is Pausiaca. Oh,
that is another question. What is a Pausiaca? Whoever had seen the type of Pausiaca and Olivacea globosa. So for now, it's a good clade. It's a good species.
Okay, good.
We can name it Pausiaca or Olivacea globosa, but we will see.
Okay.
We will see.
Okay, terrific. Thank you.
Just to comment on that, I think the phenomenon falls into two parts. First is that you have a lot of truly new species. And when we were in Australia, we did spore collecting in the herbarium in Melbourne, and now we try to barcode these things, and most of it is, yeah, absolutely. Another point is that you have species with high fruiting propensity,
right,
which have a low fruiting propensity in other parts of the world. One of these examples may be this Arteria pausiaca.
Yep.
The other one, which was completely obvious to me when I was in Tasmania, is Trichia verrucosa, which is everywhere on every log. And I asked Sarah, no, where is Trichia varia? Never seen.
No, we don't have Trichia varia in Australia.
That's amazing, you know. So
yeah,
probably, and we now have evidence, and Karina, we should do a little paper on that, just comparing barcodes to make clear that the gene flow probably between Australia and the rest of the world is that low that once in a couple of million years, probably a species can successfully invade Australia and vice versa.
Yes.
But it is probably not sufficient to prevent the things then from further speciation in Australia. Yeah? So once it has entered Australia, it develops into something amazing.
One comment and one question. When I saw the spores of the Hemitrichia non-decipiens, the spores look similar to what I have seen from Norway that I have called crateriformis.
Yes.
But macroscopically, I think they're different. They don't have the deep caliculus, the olivaceous color, and so on. So I'd really like to discuss why you said the spores were quite different. But anyway, we can discuss later.
Absolutely.
And the Perichena you showed, I mentioned to you just a while ago that I had a specimen, small specimen, sent to me from Malaysia. It looks very similar. Okay. It has a couple of spirals on the apertures. So I just want to question you why you consider it to be a Perichena with quite clear spirals. Just curious.
Why I think it's hemitrichia. It's the question is, why do I think it's hemitrichia?
No, I meant this last one, the Perichena.
Yeah.
The portrait is like.
Yeah. Well, I thought it was a Perichena, but Carlos thinks it's a could be a hemitrichia. Yeah.
And I would like to comment on also that I have the similar experience as Martin has from Australia, from the Andes of the southern South America. For instance, there was one species, the Dimium nivicola, that is very rare in the northern hemisphere. Yep. It was every fourth myxomycetes, Niviculus myxomycetes found in the Andes.
Yeah.
So there are some species which are very common in the southern hemisphere and rare in the rest of the world. So we simply should put more attention to studying myxomycetes in the southern hemisphere. I think we can find a lot of new species and also.
There's still a lot to learn. There's no doubt about it. Yeah. Okay. So I think that will do.
And a small remark about this Perichena, which is Trichia botrytis. So this year appeared a new article from Russian colleagues, which made they found out that Trichia botrytis is a complex of species, and they described like nine new species of from
Okay.
this complex.
Good.
And yes,
Yes.
some of them really remind something like that. So
Okay.
yeah.
But they won't have seen any Australian material. Yeah, it's all right. It's all right. We'll work toward that, won't we?
One small question. Were some genetic studies of the samples performed, or you're limited to studying their morphology and identified species on according to morphology?
Yes. So that's the only way I can study slime molds.